Very good evening students. So, here we are to deal with the complete syllabus of botany in our NEET World's One Step to Medicine, wherein we are going to start the syllabus from the chapter Photosynthesis and Higher Plants, wherein plant physiology and cell biology of course is weighing 50% of your NCERT 11. So, we are starting from photosynthesis and higher plants. Today we are going to deal with early discoveries, action spectra, absorption spectra, photosynthetic pigments, chloroplast, cyclic photophosphorylation, non-cyclic photophosphorylation, chemiosmotic hypothesis. Okay, so stay tuned and focus till the end of class. Fine. Anywhere, any doubt you can ask me, beta. So here, before discussing the discoveries of photosynthesis, first let us recall the standard equation of photosynthesis. Okay. So what is standard equation of photosynthesis, beta? 6CO2 plus 12H2O in the presence of sunlight and chlorophyll. It will synthesize the primary product of photosynthesis. What is that, ma? C6H12O6, which is the primary product of photosynthesis, and 6H2O and oxygen is evolved as a byproduct. This reaction is clear to you, right? Standard equation of photosynthesis. From this it is very evident that what is the raw materials of photosynthesis? Carbon dioxide, water and sunlight. These are the raw materials of photosynthesis. Primary product is glucose, byproduct is oxygen. Clear, right, ma? Here there are some synonyms for photosynthesis. You have to know that. Okay, that I will explain you clearly, taking the standard equation only as the example. Photosynthesis is also called as physico-chemical process. Okay, this is also called as physico-chemical process. Fine. This is also called as endergonic process. Okay, also called as an anabolic process. Okay, anabolic process, and this is an oxidation reduction reaction process. Both oxidation and reduction occurs. Okay, let us understand each synonym one by one. Okay, ma? So first, if you take a physico-chemical process, what is physico-chemical process? The physical energy, that is sunlight, which is absorbed in the form of photons, is converted into chemical energy, that is ATP and NADPH. Okay. So the physical energy, what is the physical energy? Sunlight, which is absorbed in the form of what? Photons, is converted to chemical energy. What is that? ATP and NADPH. This ATP and NADPH are called as assimilatory power. That is why it is called as what, ma? Physico-chemical process. Endergonic process, where light energy is being utilized in the process. Okay, endergonic means energy is being utilized for the process of synthesis of complex food molecules. And then anabolic process. What is anabolic process, ma? From simpler substances, if complex substance is synthesized, that is anabolic process. Yes or no? So the simpler substances, carbon dioxide, water and photons, okay, they are collectively making one complex product, that is glucose. Okay, that is anabolic process. And here in this one reaction, both oxidation and reduction are taking place. Okay, oxidation have, here you can see water molecule, where protons are being removed from this. Water molecule is converted to oxygen. This is oxidation, right? Removal of hydrogen or addition of oxygen is called as oxidation process. So water molecule is getting oxidized over here. If you consider the carbon dioxide is reduced, where carbon dioxide is reduced to glucose. Okay. So here what is that, ma? Oxidation reduction reaction. In oxidation you can take down as carbon dioxide, water molecule is oxidized to oxygen. Okay. Reduction, wherein carbon dioxide is reduced to glucose. Clear, right, with all the synonyms? Now you tell me Photosynthesis is also called as physico-chemical process, endergonic process, anabolic process, oxidation, reduction, reaction. Very good. Now we will look into the discoveries of photosynthesis. To understand the discoveries of photosynthesis also, we will place our standard equation of photosynthesis. Okay. So again, once again, standard equation of photosynthesis: 6CO2 plus 12 H2O in the presence of sunlight and chlorophyll. Yes, gives the primary product. What is the primary product over here, beta? C6H12O6 plus 6H2O plus 6 oxygen evolved as byproduct. So here if you have to look into, okay, from the early discoveries, what did they discover actually? Is carbon dioxide raw material of photosynthesis? Is photosynthesis taking place in the presence of water, sunlight, and is oxygen evolved from photosynthesis? Is glucose the primary product of photosynthesis? And who gave us that oxygen is coming from water, not from carbon dioxide? This and all questions are answered by the early discoveries of photosynthesis. First, primarily, who said that carbon dioxide is essential for the process of photosynthesis? That is said by Moll's half-leaf experiment. Carbon dioxide is essential for photosynthesis is given by whom? Moll's half-leaf experiment. Fine. I will be telling you, see, what is Moll's half-leaf experiment. Can you see this diagram clearly, ma? So here a potted plant is taken which is placed under the sunlight. Of course, this experiment few of you have done in your 10th standard. So see how the plant which is exposed to sunlight, half of the leaf is kept in potassium hydroxide solution. Okay, half of the leaf is placed where? In potassium hydroxide solution. So in this half leaf, whichever carbon dioxide is there, that is completely absorbed by KOH solution, and this gets converted to potassium carbonate. Okay. So half leaf which is kept in KOH solution, the carbon dioxide is completely absorbed and it is converting to what, Nana? Potassium carbonate. Potassium carbonate it is converted, and rest half leaf which is freely exposed to sunlight is happily conducting the photosynthesis. Okay. So after certain period of time, when this leaf is conducted with starch test, half of the leaf which is exposed to sunlight has shown the blue black color, indicating the presence of starch. Okay. When starch is reacted with iodine, it will show what color? Blue black color. Okay. But half of the leaf which is kept in the KOH solution has shown you the pale color, indicating the absence of starch. Okay. So here carbon dioxide is not there, so fixation is not happening, glucose formation is not happening. Okay. Here it is there, so glucose formation happened. So this is the experiment of what? Moll's half-leaf experiment. Clear, right, beta? So here what you have to remember, carbon dioxide is essential for the process of photosynthesis is said by Moll's half-leaf experiment, wherein he has used KOH solution. Okay, and half of the leaf which is exposed to sunlight has turned to blue black color, half of the leaf which is kept in the KOH solution has shown the pale color, indicating the absence of carbon dioxide. Clear, right, ma? Now tell me which scientist has told you the water, sunlight are essential for photosynthesis and oxygen is evolved from photosynthesis. That is being told by Jan Ingenhousz. Okay. He is the scientist who has conducted experiment on hydrilla experiment. Okay. So hydrilla plant, the experiment is conducted and he said that water and sunlight are essential for the process of photosynthesis and oxygen is evolved from photosynthesis. Let us look at that experiment as well. So nothing like a beaker containing, yes, can you see this, ma? A beaker containing water is taken, a distal funnel inside which the hydrilla plant is placed. Okay. This distal funnel is inverted into the beaker containing water. Over this distal funnel, the test tube is placed like this. Over the distal funnel, test tube is placed. Okay. Two such setups are made. One is kept in sunlight, one is kept in dark. Okay. The setup which is kept under sunlight, after certain period of time, some water bubbles are appeared over this test tube, indicating the presence of oxygen. Whether it is oxygen or any other bubbles is identified. How did they identify that? They have put a burning flame over the test tube before opening that has burnt more vigorously. The flame has burned more vigorously, indicating the presence of oxygen. Clear, right, ma? So, this hydrilla experiment is conducted by whom? Jan Ingenhouse. Okay. Hydrilla experiment is conducted by Jan Ingenhouse, wherein what did he say? Water, sunlight are essential for photosynthesis and oxygen is evolved from photosynthesis. So, take down. Jan Ingenhouse has said that water and sunlight are essential for photosynthesis. Oxygen is evolved as a byproduct of photosynthesis. Okay. Next, who said that glucose is the primary product of photosynthesis? This is given by the scientist von Sachs, who has conducted experiment on variegated leaf. What is variegated leaf? Okay. Have you ever seen leaf in different colors? Okay. Have you ever seen a leaf with some green parts and some non-green parts? Yes, right. There are some leaves as such. Those leaves are called as variegated leaves. Okay. Or else if a leaf is taken, complete green leaf is taken and half of it is covered with some black paper. Such leaf exposed to sunlight after certain period of time, when it is conducted with starch test, this part will show blue-black color, whereas this region which is covered with black paper will show pale color, indicating the absence of starch. Okay. So, this is the variegated leaf experiment conducted by whom? Von Sachs, and he said that glucose is the primary product of photosynthesis. Now, let us come to oxygen. Who discovered oxygen? Oxygen discovery is by Joseph Priestley. So, this scientist Joseph Priestley has started working on the discovery of oxygen since 1770s. He has conducted series of experiments using mice and bell jar. So, what did he do? A bell jar inside which he has put a mouse and a candle, burning candle. After certain period of time, candle has put off and mouse has died. But the same experiment, when it is repeated with a mint plant, okay, with a mint plant, mouse and a burning candle, it was the same even after certain period of time, with which he has concluded that whichever gas is respired out of the plant is taken by the burning candle and the animal, and whichever is expired by this is taken by the plant. Okay. So, with this he said that gas is what? Oxygen. As such, oxygen discovery is done in the year 1774. Now, it is fine. Okay. Oxygen is discovered by, yes ma'am, Joseph Priestley. So, it is clear to you, right? Oxygen is discovered by Joseph Priestley. Oxygen is evolving from photosynthesis is said by Jan Ingenhouse. But then there is one more scientist who says that till here everybody thought that oxygen is coming from carbon dioxide. But it is C.B. Van Niel who has conducted bacterial photosynthesis and said that oxygen which is evolved from photosynthesis is coming from water, not from carbon dioxide. Okay. Wherein Who is that scientist? C.B. Van Til. What did he do, ma? He has conducted bacterial photosynthesis. What is bacterial photosynthesis? Anoxygenic photosynthesis, where the electron donor will be H2S. Okay, electron donor will be what? H2S mostly. So, from here when it is taken, sulfur molecules are evolved as byproducts. In that case, in oxygenic photosynthesis, water molecule is the electron donor and it is releasing out what as byproducts, ma? O2. That's it. Okay. So, who said that? Oxygen is coming from water, not from carbon dioxide. C.B. Van Til, by conducting what photosynthesis? Bacterial photosynthesis. Clear, right? Any doubt anywhere? No, right, ma? So, here Jan Ingenhousz, who said that sunlight is essential for the process of photosynthesis, he didn't mention which spectrum of sunlight is actually essential for this process. Okay. So, the visible spectrum of sunlight, which ranges from 300 nanometers to 700 nanometers, from that visible spectrum of sunlight, photosynthetically active radiation is required for the process of photosynthesis, wherein maximum amount of photosynthesis is done in pi. Okay. So, which wavelength of photosynthesis is actually necessary for this process is explained by E.W. Engelmann. Okay. Who is that scientist? E.W. Engelmann. What did he do? Which spectrum of sunlight is essential for photosynthesis? Which spectrum of sunlight is essential for photosynthesis? So, usually you have to be clear with this. The visible range of sunlight is 300 to 700 nanometers. Of that, photosynthetically active radiation is 400 to 700 nanometers. So, he has conducted one nice experiment in quite a times of your NEET previous year questions. 1 or 2 times this question has appeared. Okay. So, listen to this carefully. Drop your pens, listen with attention. So, what did he do, ma? He exposed unilateral sunlight to a prism. Okay. When unilateral sunlight is exposed to a prism, the light gets scattered to VIBGR like this. Okay. It gets scattered to VIBGR. Fine. This scattered light is exposed to a filamentous green algae, that is Cladophora. So, here what is the model organism? Cladophora. Of course, Cladophora is a filamentous green algae. It is conducting what type of photosynthesis, ma? Oxygenic photosynthesis. Oxygenic photosynthesis. When it is conducting oxygenic photosynthesis, it will release oxygen as a byproduct, right? So that above this filamentous green algae, aerobic bacteria are placed. Okay, above the filamentous green algae, what are placed? Aerobic bacteria. Okay. After certain period of time, he has observed that aerobic bacteria got accumulated in blue and red regions of light. Okay, where did they accumulate? Blue and red regions of light. Once again, I will repeat the experiment, listen with attention. Unilateral sunlight is exposed to prism. Then it scattered to what? VIBGR. This scattered light is exposed to filamentous green algae, that is Cladophora. It is conducting what photosynthesis? Oxygenic photosynthesis. Above this, aerobic bacteria are placed. After certain period of time, he has observed that aerobic bacteria got accumulated more near blue and red regions of the light. Okay. With this, he has given the action spectra and absorption spectra. Action spectra and absorption spectra. Okay. Absorption spectra. Clear, right ma? Fine. So here if you see, yeah, from the standard equation of photosynthesis, once let's recall the complete discoveries. Who said that carbon dioxide is essential? Mole's half-leaf experiment. What chemical did he use? KOH. Half of the leaf has turned to pale color, half of the leaf blue-black color after the starch test. Okay, second, which scientist said that water and sunlight are essential for photosynthesis and oxygen is evolved as byproduct of photosynthesis? Who said that? Jan Ingenhousz. Okay, oxygen is evolved as byproduct. And who discovered oxygen? Joseph Priestley. Who said that oxygen is coming from water, not from carbon dioxide? C.B. Van Neel. Who said that glucose is formed as a primary product of photosynthesis? Von Sachs. By what experiment? Variegated leaf experiment. Yeah, it is well and fine. Jan Ingenhousz has said that sunlight is essential for photosynthesis. But which spectrum of sunlight is efficient for photosynthesis is said by E.W. Engelmann. Okay, wherein after this experiment he has drawn out what? Action spectra and absorption spectra. Clear with all the discoveries and all the experiments, ma? Shall we move on to action spectra and absorption spectra? Yes, right? Before learning action spectra and absorption spectra, you should clearly know about photosynthetic pigments. Okay, photosynthetic pigments. Where are these photosynthetic pigments present actually? Okay, so for example, let me take a leaf like this. Okay, leaf is composed of what cells? Mesophyll cells. Yes or no? Leaf has what cells, ma? Mesophyll cells. Mesophyll cells will be having organelles. What organelles? Chloroplast. Fine. We know chloroplast is a double membrane bound organelle. It has what? Sac-like structures, thylakoids and grana. Membranous sac-like structures, thylakoids. Stalk of sac-like structures are grana. They are attached by what? Stroma lamellae. They are connected by stroma lamellae. No issues. So inside this fluid filled space is what? Stroma. Okay. So thylakoids, stroma lamellae and stroma, fluid filled space inside the chloroplast. Clear, right? So this photosynthetic pigments are present in this membranous sac like structure. Okay, in the thylakoid membrane, if this is the thylakoid membrane, here the photosynthetic pigments are present. Okay, inside the thylakoid membrane there are photosystems. Inside photosystems you will have light harvesting complexes. Inside light harvesting complexes, photosynthetic pigments are present. You got the location of photosynthetic pigments? Okay. So leaf will have what cells, ma? Mesophyll cells. Mesophyll cells will have what organelles? Chloroplast organelles. Each chloroplast is a double membrane bound organelle. Inside each chloroplast you have membranous sac-like structures. What are they? Thylakoids. In the membrane you will have, in this membrane you will have photosystems. Okay, inside photosystems you will have light harvesting complexes. Inside light harvesting complexes you have what? Photosynthetic pigments. Okay, as such you have different photosynthetic pigments which absorb light at different wavelengths. Okay, so that photosynthetic pigment which absorbs light at maximum wavelength is called as core pigment. Okay, those pigments which absorb light at minimal wavelengths are what? Accessory pigments or antenna pigments. Till here it is enough to understand action spectra or absorption spectra. Later I will explain more elaboratively. Okay, so here most of the leaves appear green in color because they reflect the green color. Okay, and they absorb most of blue and red colors. Okay, blue and red regions of light are mostly absorbed and they will reflect what? Green light. Okay, that is why most of the leaves will appear to be green in color. Okay, understood. So as such, when in chromatogram these different pigments are viewed, each pigment has its own color. Okay, chlorophyll A is what color, ma? Chlorophyll A is blue green in color. Blue green in color. Chlorophyll B will be yellow-green in color. Okay, carotenoids are yellow-orange, yellow to orange or orange color. Fine. And xanthophylls are yellow color. Xanthophyle is yellow color. Clear? Now, action spectra and absorption spectra. Look at this carefully. First, you have to know the difference between action spectra and absorption spectra. Absorption spectra will depict you, okay, different pigments absorbing light at different wavelengths. Okay, different pigments absorbing light at different wavelengths is seen where in the absorption spectra. In action spectra, maximum amount of light is absorbed by which pigment at the maximum wavelength, and maximum oxygen is evolved at that wavelength is shown by what? Action spectra. Simple beta. In absorption spectra, different pigments will absorb different wavelengths of light, that is predicted. In action spectra, one pigment which is absorbing maximum wavelength of light and evolving maximum oxygen, that is being plotted in the action spectra. Okay. So, now you look at the absorption spectra first, compare it with the action spectra. So, here in our NCERT, how many pigments are given in the absorption spectra? 3 pigments are given. Okay. First is what? Chlorophyll A, then is what? Chlorophyll B, then carotenoids. Let us plot the wavelengths, so it will be clear. Here 400 nanometers, 500, 600 and 700. Okay. PAR, photosynthetic active radiation is given. Now you see chlorophyll A, it shows 2 peaks, one in between 400 to 500 nanometers and one in between 600 to 700 nanometers. 2 high peaks are being shown. Second, from 450 to 500 nanometers, one long peak is shown by chlorophyll B, one very small peak in between 600 to 700 nanometers. Same the case with carotenoids, 2 peaks are observed between 400 to 500 nanometers. Amongst these 3 pigments, it is said that chlorophyll A is absorbing maximum wavelength of light. Why? If you can say it is fine, if not I will be telling you. Amongst these 3, why chlorophyll is considered as the maximum light absorbing pigment? Should I see? Here can you see 2 high peaks, one in between 400 to 500, one in between 600 to 700. Can you see any other pigment showing the same intense peaks? No. Okay. Here it is very small negligible peak, we cannot consider that. Here 2 peaks in one wavelength only, one slot of wavelength only. That is the reason, as it is showing maximum wavelength of light, maximum absorption at 2 different wavelengths, at both blue region and red region of light. Okay. So, that is why chlorophyll A is said to be the core pigment or reaction center which is absorbing maximum wavelength of light. In action spectra, when it is plotted, chlorophyll A of absorption spectra is coinciding with the chlorophyll A of action spectra. So, look at here beta ones, maximum absorption, these are the direct liners of your NCERT, maximum absorption by chlorophyll A, that is in blue and red regions, also show higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. Clear, right? So, till now what did we learn? The synonyms, all the synonyms of photosynthesis with explanations. We have completed the discoveries with each experiment explained in detail, and then now we are clear with action spectra and absorption spectra. Okay. Next. Now, let us look into the chloroplast organelle. Okay. You specify beta, you specify which region you didn't understand, that I will explain again. Okay. Specify it clearly. Chloroplast. So, we all know chloroplast is what organelle? double membrane bound organelle. It is very evident from here, right? So, this double membrane bound organelle inside which the fluid filled matrix is called as what? Stroma. This membranous sac like structure is called as what? No, thylakoids. Membranous sac like structure inside the chloroplast is thylakoids. Okay. The stalk of sac like structure is grana. Okay. Stalk of sac like structure is what? Grana. Membranous sac like structure is thylakoid. Stalk of that sac like structure is grana. One grana is connected to the other grana by what? Stroma lamellae. One grana is connected to other grana by stroma lamellae. Clear till here, right? Now, in chloroplast and mitochondria, these two organelles are popularly known as semi-autonomous organelles, semi-autonomous organelles, or self-duplicating organelles, Fine? Okay, chloroplast is called as semi-autonomous or self-duplicating organelle because they have double-stranded circular DNA. Double-stranded circular DNA. What is the ultimate goal of DNA? Ultimate goal of DNA is to synthesize protein. As such, the double-stranded circular DNA, whichever is present in the chloroplast, will synthesize the world's abundant protein, that is rubisco. Okay, clear till here? Okay, now, and they have ribosomes. Ribosomes whichever are present inside the chloroplast, they are 70S ribosomes. Clear, right? So, this is the complete description of the double membrane bound organelle chloroplast. Any doubt anywhere? No, right? Yes. Next.
In ribosome 70 is present.
Yes, 70S ribosomes are present whose larger subunit is 50S and smaller subunit is 30S. Okay. Next. So, now I told you, right, first let us understand the core pigment and accessory pigments in detail, then it will be easy for us to understand the cyclic photophosphorylation, non-cyclic photophosphorylation. Okay. So, look at here carefully, this is the diagram again given in your NCERT. Okay. So, from this double membrane bound organelle, that is chloroplast, these are what? This is what? Grana. This is what? Thylakoid. Okay. One thylakoid I am taking here and drawing. Okay. Okay. This inside region is called as thylakoid lumen. Okay. This is called as thylakoid membrane or thylakoid lamellae. Fine. This is thylakoid lumen and this is thylakoid membrane or thylakoid lamellae. I told you, right, inside the thylakoid lamellae there are photosystems, PS2 and PS1. These are placed one after the other based on their discovery. Okay, first PS2 is discovered, then PS1. Inside this photosystems, what are present? I told you, light harvesting complexes. Okay, each light harvesting complexes will have antenna pigments and core pigments. Core pigment. Antenna pigments are also called as accessory pigments. Antenna pigments are accessory pigments. Fine. Antenna or accessory pigments include what? Chlorophyll B, carotenoids and xanthophylls. And this you see, core pigments include what? Chlorophyll A. Clear right now. So, you have what? Inside the thylakoid membrane, photosystems, Inside photosystem you have light harvesting complex. Light harvesting complex will have antenna pigments and core pigments. Core pigment chlorophyll A, antenna pigments or accessory pigments will have chlorophyll B, carotenoids and xanthophylls. Clear, right? Any doubt anywhere? No, right ma? So now here you see chlorophyll A and B structure. What will be the structure of chlorophyll? A porphyrin ring and a phytol chain. A porphyrin ring like this and a phytol chain. This porphyrin ring, these carbon units are connected to the magnesium which is present at the center. Okay. So, chlorophyll structure has what? Porphyrin ring and phytol chain. So, this core pigment, that is chlorophyll A, is also called as reaction center. Clear? Till here you have understood now? If this is clear to you, then you will easily understand non-cyclic photophosphorylation. Okay? So, here you see, before entering into the non-cyclic photophosphorylation, once let me clearly tell you, once let me clearly explain you like what is the sequence of photosynthesis that is happening. Okay? Photosynthesis is a sequential process of light reaction and dark reactions. Okay? Let us first understand this sequential process. Take down this table as it is, ma. Light reactions, also called as light independent reactions. These are dark reactions or light independent reactions. Got it? So, here light reactions, there are three sequential steps. Today we are going to learn this. First is absorption of light in the form of photons. Absorption of light in the form of photons. Second will be photolysis of water. Photolysis of water. And third is formation of assimilatory power. Formation of assimilatory power. This assimilatory power can be ATP and NADPH. Okay? Utilizing this assimilatory power, reducing carbon dioxide or fixing carbon dioxide to glucose is dark reaction. Carbon dioxide fixing to glucose. Clear? Now, sites or locations of each of this is very important. So, first if you see absorption of light in the form of photons, where is it happening? Inside the thylakoid membrane by photosystems, by photosynthetic pigments, light is absorbed in the form of photons. So, this location is where ma? Thylakoid lamellae. Okay? Utilizing this photons, photolysis of water takes place inside the thylakoid lumen. I told you, right, if you consider this as thylakoid membrane with photosystems, here photolysis of water take place. Okay, wherein one water molecule is lysed to 2 protons plus 2 electrons plus oxygen atom. Okay, so this takes place where in the thylakoid lumen. Fine. And formation of assimilatory power, that is ATP and NADPH, are formed in stroma. Okay, stroma. This energy is being utilized and carbon dioxide is fixed to glucose. This fixation process utilizing the assimilatory power, okay, This occurs again in the stroma. Okay. Here you can expect a statement based question wherein light independent reaction, calling light independent reactions as dark reaction is a misnomer, because dark reaction means it has to take place in dark, but no, it occurs in daytime also, but then this is not directly dependent on light, it is dependent on the products of light reaction. Okay, it is dependent on what? Products of light reaction. So, till here everything is clear to you, na? Okay. So, shall we continue here or let us learn this tomorrow? Your wish. If you are interested, I will be continuing. If not, tomorrow I will discuss. Continue? Okay, fine. Listen. Again, a thylakoid. Photosystem. Okay. First step is what? Absorption of light in the form of photons. Inside this photosystems, what are there? Light harvesting complexes, and inside that pigments are present. So, here they are absorbed in the form of photons, right ma? Now, this energy, this photons energy is being utilized by the water molecules whichever are present in the thylakoid lumen. This is thylakoid lumen and this is what? Stroma. Understood? So, this photons energy is utilized by this water molecule and this gets lysed to what? 2H plus 2 electrons plus oxygen atom. Okay. So, here there is one complex present attaching to the photosystem 2 and thylakoid lumen. Attaching to photosystem 2 and thylakoid lumen, there is a complex present. What is that? Oxygen evolving complex. Attaching to this oxygen evolving complex, water molecule is lysed and oxygen is evolved inside the thylakoid lumen. Till here it is clear? Any doubt anywhere? Okay. So, next the first step absorption of light in the form of photons, and second step is clear to you know. Now, third step, that is formation of assimilatory power. To understand that, you have to understand this non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Okay. This non-cyclic photophosphorylation will occur in oxygenic photosynthesis. Wherever oxygen is liberated, their non-cyclic photophosphorylation will occur in oxygenic photosynthesis. Okay. So, here if you see ma, okay, no issues. Let us continue. So, non-cyclic photophosphorylation will occur in oxygenic photosynthesis. Cyclic photophosphorylation will occur in anoxygenic photosynthesis. So, till here you make a note of it. Cyclic and non-cyclic photosphorylation I will tell you tomorrow. Okay. So, till here everything is clear, right? Absorption of light in the form of photons and photolysis of water. Formation of assimilatory power and dark reactions I will begin tomorrow. So, in today's class what did we learn ma? First, synonyms of photosynthesis. Photosynthesis is also called as physico-chemical reaction. Why? Because photons, physical form of energy, that is photons, is converted to chemical form of energy, ATP and NADPH. Second is, it is also called as endergonic process. Third is anabolic process, because it from the simpler molecules complex glucose molecule is being synthesized. Oxidation reduction reaction happens, wherein oxidation is water molecule getting oxidized to oxygen, reduction carbon dioxide is reduced to glucose. Clear, right? Clear with all the synonyms. Next is what ma? Our discoveries. Carbon dioxide is essential for the process of photosynthesis said by Moll's half-leaf experiment. Water and sunlight are essentials. Jan Ingenhouse. Who said that oxygen is evolved from photosynthesis? Jan Ingenhouse only. Oxygen evolved as byproduct is said by Jan Ingenhouse. Who discovered oxygen? Joseph Priestley. Who said that oxygen is coming from water, not from carbon dioxide? C.B. Van Neel. What photosynthesis he conducted? Bacterial photosynthesis. What is the electron donor in bacterial photosynthesis? H2S. In oxygenic photosynthesis, what is the electron donor? H2O. Glucose is the primary product of photosynthesis said by Von Sachs. What is the experiment he conducted? Variegated leaf experiment. Clear, right? Who said that? Which spectrum of sunlight is essential for photosynthesis? E.W. Engelsmann experiment. Who gave us the action spectra and absorption spectra? Who gave us action spectra, absorption spectra? E.W. Engelmann. Okay. And then about photosynthetic pigments. Where are photosynthetic pigments present? In chloroplast, thylakoid membrane, inside the light harvesting complexes. Locations are very much important. Sites where the reactions are happening, very much important. Okay. And then action spectra and absorption spectra. In absorption spectra, we have different pigments, chlorophyll A, chlorophyll B, carotenoids absorbing light at different wavelengths, in which chlorophyll A absorbs maximum wavelength of light. And then action spectra, it is only chlorophyll A. And here from the chromatogram, we got to know the colors of the pigments. Chlorophyll A is what color, Nana? Blue-green color. Chlorophyll B is what color? Yellow-green in color. Carotenoids of what color? Yellow-orange color. Xanthophylls are of what color? Yellow color. Clear, right? Next. Chloroplast structure. Double membrane bound organelle. Membrane sac-like structure is thylakoid. Stalk of sac-like structures? Grana. One grana connected to other by stroma lamellae. Inside this stroma, ribosomes are present. What ribosomes? 70S ribosomes. Starch granule and lipid droplets are also present. This is called as semi-autonomous organelle or self-duplicating organelle by because they have what? Double-stranded circular DNA, wherein it has the ultimate goal of this DNA is to synthesize protein, that is rubisco. Okay. Now, what are the accessory or antenna pigments? Chlorophyll B, carotenoids and xanthophylls, where they will absorb light at different wavelengths. Chief pigment or core pigment is chlorophyll A, okay, wherein chlorophyll A is also called as reaction center, where this absorb light at different wavelengths and submit it to what? Core pigment, that is chlorophyll A. This is present in the light harvesting complex of photosystem in the thylakoid membrane. Clear, right? Next. These are the sequence of the reactions of photosynthesis, okay, of which till here it is clear to us. Assimilatory, formation of assimilatory power we will look into tomorrow, and arc reactions as well. Okay. Clear right now? That's all for today, and thank you. Don't miss the session at all. We are going to cover the complete syllabus in the most detailed way. Okay. Thank you.